first speaker here is Dr. Kim Green, and Dr. Green did his earlier studies in the UK. You'll figure that out as soon as he opens his mouth. And from the University of Birmingham, and then at Leeds. And after that, he came here to UCI MIND, and he has started his own lab in 2011. And he works on a particular cell type called the microglia, which is near and dear to my heart because it's involved in inflammation and immunology. And he's been studying the role of microglia in Alzheimer's disease. Now, he's published in his young career already over 60 papers in peer-reviewed scientific journals, and he has funding from the NIH and the Alzheimer's Association, the Whitehall Foundation, very prestigious, and the American Federation for Aging Research. And so he is really astute and really cares about the work that he has been doing, wants to understand more of the risk factors such as stress and inflammation. And so Dr. Green, come up and entertain us here.
All right, thanks to the speakers for inviting me to talk, and thanks to you guys for sticking around to listen to what we've got to say over here on the research side of things. All right, so today I'm going to be telling you about research that my lab has been conducting over the past few years into cells known as microglia. I'll be telling you about what these cells do and why we're interested in studying them. So here's a picture of some microglia. Microglia are basically the immune cell of the brain. So they function much like the white blood cells that you see in the rest of the body, except you find them in the brain. This is what they look like. They have this nice big cell body, and then they have these processes that radiate out. And what you see is they're nicely positioned all over the brain, spaced evenly out from one another. And what they're doing is they are constantly surveying the local brain environment. These processes are constantly feeling around, looking for stuff to do. And primarily, their function is to protect us from infections in the brain. So these microglia are looking around, and if they find evidence of a bacteria, a virus, some sort of pathogen, they will then respond to it. They will activate, as we say. They will mount an inflammatory response. They will then produce cytotoxic chemicals designed to kill these pathogens and get rid of them from the brain. Now, this is a great thing for them to do. You know, they're very useful to us. But we're interested in them because they are also implicated in pretty much every brain disorder that there is. For example, they're heavily implicated after traumatic brain injury, where they become a little bit too overactivated and actually impede the recovery. Likewise, they're implicated in aging, where it's thought that they contribute to age-related cognitive decline. And finally, they're implicated in every single neurodegenerative disease, including Alzheimer's disease, but also Parkinson's, Huntington's, ALS, MS, anything you can think of, these cells are implicated. And so it's really important that we study them to understand their biology and find ways to control them. Because if we can, then we may be able to find a way to treat all these various different disorders that involve inflammation in the brain. So let me just show you something. These microglia, when they respond to a pathogen or damage in the brain, they become activated. And basically, I don't know if you can see this, but there's a series of red dots here, each one of which is the cell body of these microglia. Over here we have a mouse that has undergone a lesion. This is similar to a bacterial infection, and all of the microglia have become activated. And you can hopefully quite clearly see that these cells look different. They've grown in size and they've become more numerous. At this point, they are secreting all of these nice cytotoxic chemicals designed to kill off infections and everything else. And once they've done their job, they then resolve. They then revert back to this normal state. So this is all very helpful. They respond to infections, they respond to damage, and once they have cleared away the problem, they then revert back to normal. But what I'm going to show you now is what happens in diseases like Alzheimer's disease, where these cells go a little bit wrong. And so this brings us to the Alzheimer's brain. Now, you've all seen this slide before, and this just illustrates that the Alzheimer's brain is a lot smaller than the brain from a normal person. And if you were to weigh it, it would weigh about a third less. And the reason for this is that over the course of the disease, many of the brain cells, the neurons, have died off. And of course, these are the cells that store our memories, that form connections with one another to give rise to our personalities, our traits, etc., etc. And so one of the key questions is, well, why have these cells died off during Alzheimer's disease? What has caused this? Well, you've all seen this as well. If we look inside the Alzheimer's brain under a microscope, we can see several hallmark pathologies. Primarily these extracellular plaques, which are composed of this amyloid beta peptide, and then these intracellular tangles, which are composed of this tau protein. And the net presence of these two pathologies in the brain gives rise to synaptic and neuronal dysfunction, which over time leads to the neurons dying and gives rise to Alzheimer's disease. But there's one other highly prominent feature of the Alzheimer's brain, and that is inflammation. You have plaques, you have tangles, you have neuronal loss, but you also have a chronic, ongoing inflammatory process throughout the entire course of the disease. So, for example, these are human brains. This is PET imaging here, and we can physically look at inflammation in human brains on a scanner. And this is just a normal brain, and you can see that there isn't too much activity going on there. But if you look inside the brain of someone who has Alzheimer's disease, these hotspots are all areas where there is pretty elevated inflammation going on. So why is this? Well, what happens is that in the Alzheimer's brain, these plaques form. And the microglia suddenly see these plaques for the first time. And they're a little bit confused. They've never seen these plaques before. And they respond to them because they think that they are a pathogen. They think that they're bacteria. So they mount an immune response against these plaques. They migrate towards them. They surround them. They then start to produce all of these cytotoxic chemicals designed to kill pathogens. But the problem is that the plaques are not alive. They're not going to be killed off by all of the things that the microglia are doing. But the microglia don't know this, they don't give up, and so they stay activated, trying to do something about these plaques for the whole course of the disease. And so now we not only have these plaques, but we have our own microglia, which are trying to help, but in actual fact are now damaging the local brain environment. So let me just show you a picture of this. These are from mice. Up the top is just a normal, healthy mouse. And each one of these red dots that hopefully you can see is the cell body of a microglia. Down here we have a mouse that has been genetically engineered to develop Alzheimer's disease. And as it ages, it develops plaques in its brain, which are shown here in green, these lovely crater-looking type things. Over here is the same microglia stain, and you can really see that they have now become highly activated and they are surrounding those plaques. This is because they're trying to help, but unfortunately they're unable to do so. And so now, because of this, the Alzheimer's brain has this chronic, ongoing inflammatory response mediated by microglia that will last for the whole course of the disease. And as I said, this is going to be harmful to the local brain environment. This is going to contribute to the synaptic loss and the neuronal loss that you see in the Alzheimer's brain. And this is the same thing that you see in all of these other neurodegenerative diseases as well. You get this chronic activation of these cells in Parkinson's disease, Huntington's disease, multiple sclerosis. Pretty much anything you can think of, you get a similar thing occurring. And so this is why it's so important for us to be able to study the biology of these cells and to find ways to be able to control them. So to that end, my lab has been focusing on a receptor known as the colony-stimulating factor 1 receptor. I'm going to call this the CSF1R. And in the brain, this is only found on microglia. But in the rest of the body, it's expressed on similar cells. And those are monocytes, macrophages, dendritic cells, and osteoclasts. It's not so important, but this receptor has two endogenous ligands, CSF1 and IL-34. And most of the functions of this receptor have been previously worked out in macrophages. Macrophages are much like the microglia of the brain, except they're found in the rest of the body. And it's known that this receptor is absolutely essential for macrophages to be able to divide in order to proliferate. So we wanted to ask the question of what would happen if we were to manipulate this receptor, this signaling pathway in microglia, and whether or not this could be useful for things such as Alzheimer's disease. And we decided to use a pharmacological approach. That means we wanted to use drugs that would inhibit this receptor to see what would happen. And we're very fortunate because a number of drug companies, biotechnology, pharmaceutical companies, have already manufactured and made inhibitors of this receptor. And so we could screen them to find out which ones would be most appropriate for what we wanted to do. And so down here is just some data. This is just us screening a bunch of available different CSF1R inhibitors to find which ones were the best at inhibiting microglial proliferation just in a dish. And we found this guy here, PLX3397 was the most potent. The lower the value, the more potent it is at inhibiting the proliferation. This guy's a broad spectrum inhibitor. It inhibits loads of different things. This one is actually very specific. In fact, it only inhibits this CSF1R, but also a related receptor known as c-KIT. Another really useful thing about this particular compound is that we found that it crossed the blood-brain barrier. And this is very, very important when you're trying to manipulate things that actually occur within the brain. So it's PLX3397, and what's also kind of neat is that it's been through clinical trials for various oncology indications, and is currently entering phase 3 clinical trials for something known as pigmented villonodular synovitis, which is a disorder of the joints. Because it's in humans already, it means that things we do here might someday be applicable to things such as Alzheimer's disease. So what I'm going to do is just show you what happens if we treat normal, healthy mice with this CSF1R inhibitor for either 3, 14, or 21 days. It's a very big effect. It's not at all what we predicted would happen. This is one of those unexpected findings in science. So here we have a brain section of a mouse. This is just a normal mouse. This structure here is known as the hippocampus. This is the area of the brain that's important for forming your memories. It's also the area that's heavily attacked in the Alzheimer's brain. And on here we have a stain for microglia, and I hope that you can see this, but each one of these black dots all over the place is the cell body of a microglia. So you can see they're all over the brain, they're spaced nice and evenly out from one another, and those towards the front can hopefully see some processes radiating out from these small black dots. So now I'm just going to show you what a similar mouse looks like after 3 days of treatment with the CSF1R inhibitors. Okay, there we go. So hopefully you can notice some pretty big changes just in 3 days. If you look, there's actually far, far fewer cells or microglia than we had seen in the control mice. In fact, if you were to count the number of black dots here, there'd be about 50% that we had in the control animals. And what's more, you can clearly see that they look different. Their processes are a little bit thicker, their staining is a little bit darker. This is what the brain looks like after about 14 days worth of treatment. Now, there are far, far fewer cells. In fact, there's only about 20% of the cells here that we found in the controls. And by this point, it's very obvious that these cells have a different morphology. They are staining much darker. You can see that they're bigger, their processes are short and stubby. So we've had some very significant changes here just within a couple of weeks. Finally, this is what the brain looks like after about 21 days. This is exactly the same microglial stain. And basically, by this point, there are no microglia left in the brain. There's a couple, here's one, that has survived. But for the most part, what we have done is cleared out all of these cells from the brain, which is pretty much unheard of and very unexpected for us. So this is just to illustrate this point a little bit further. This is just a whole brain section through a mouse brain. What we've done is we've superimposed a white dot onto each and every microglia in the brain. Over here we have a mouse that we've treated for 3 weeks with these CSF1R inhibitors, and you can see that now we literally only have a couple of handfuls of these cells left. So this is fairly unprecedented. This is basically clearing out entire tissue from the brain. This is just a quantification of that. After about 3 days of treatment, we get rid of about 50% of the microglia. After 7 days of treatment, about 70% of the cells are gone, and sometime between 7 and 21 days, basically 95 to 99% of the microglia are no longer present in the brain. So this is really remarkable. This is very, very unexpected, and this was very interesting to us because these microglia, of course, are so negatively implicated in all of these various neurodegenerative disorders. So could we perhaps begin to apply this approach to some of these diseases? So what happens to these cells? What happens to the microglia when we treat? Well, they die, and we can see this through a variety of different approaches. Firstly, we can see remnants of microglia throughout the treated brain. You can just see the remains of these cells, as you see here and here and here. Over here we have a microglia that is expressing green fluorescent protein. It glows green. And this particular cell here, we have caught it in the middle of disintegrating. These are supposed to be nice continuous processes, but instead you can see that they are basically disintegrating into a series of blobs. This is a microglia undergoing cell death. And for those who are interested in this, we have an astrocytic stain in the background, and we can actually see the GFP being taken up into those astrocytes. Additionally, we see that microglia begin to stain for active caspase 3, which is a very good indication that they are going through cell death pathways. So just to conclude, this was the finding that we made. This basically tells us that microglia in the adult brain are fully dependent upon signaling through this CSF1R signaling pathway for their survival, and that administration of drugs that inhibit this receptor that get into the brain lead to this rapid elimination of microglia throughout the CNS. And that means throughout the brain and throughout the spinal cord as well. These cells undergo cell death, and what's very interesting is that peripheral cell populations are not depleted. So the microglia are uniquely sensitive. This is not depleting the peripheral monocyte and macrophage populations. So because microglia are the only cell type within the CNS to actually express this receptor, it makes it an incredibly useful tool for now being able to work out the function of microglia, not just in the healthy brain, but in these various disease processes. But not only that, if this works out, could this potentially be a therapeutic target? Can we eliminate microglia during the disease process and bring about some sort of functional recoveries? So before we can address those questions, we have to really ask whether or not getting rid of an entire tissue from the brain is a good thing. These cells are up there for a reason. They have jobs, and we are getting rid of them all, and perhaps this is going to have bad effects on its own. Perhaps you need microglia for the way your brain functions normally. Perhaps you need them for learning and memory, for motor function, for various behaviors. And so, in order to develop this further, we have spent a lot of time looking at the behavior of mice that no longer have microglia. And I'm going to show you that data now. And the data I'm going to show you comes from mice that were 2 months of age, and then they were treated for a further 2 months. So this means they have lacked microglia for 2 whole months, which is a pretty long time when you're talking about a mouse experiment. Okay, let's see if this works. We have some videos. Okay, so I'm first going to show you a couple of tests of anxiety-type behaviors. So the first one is known as elevated plasmase, and it looks like this. It has 2 closed arms and 2 arms out in the open. And basically, we put the mouse on this plus shape, and a normal, happy, healthy mouse should spend more time in these closed arms and not venture out into the open part of the maze. Mice like to feel protected, they like to be away in little burrows, and so if it's behaving normally, it should be in this region. The second test I'm going to show you is known as open field. This is where we put a mouse into a box, and we just videotape it for a couple of minutes. And again, a mouse that is behaving normally should spend most of its time around the perimeter, hugging the wall of the container. They shouldn't want to go out into the center of the container because being out in the open is where mice can be got by predators. Hawks can swoop in and stuff like that. So let me show you what happens. It's pretty simple. The white bars are the control, the black bars are the mice that do not have microglia and have not had microglia for 2 whole months. And what's pretty obvious, doesn't matter what the measures are here, they are exactly the same. So despite the fact that these mice have no microglia, their behavior on this elevated plasmase is identical to the intact animals. Over here we have the open field. Again, we're measuring whether or not they're in one zone or the other zone, and you can see that there are no differences. So despite the fact these animals have no microglia and have had no microglia for 2 months, they have no changes in anxiety-type behaviors, which is very unexpected, but also pretty cool. Okay, next, motor function. Perhaps you need microglia to be able to coordinate movement properly. So we can test this through a couple of ways. Again, we can use this open field test. Basically, we can measure how far these mice will move within a given period of time, and we can measure how fast they move. The next test we use is known as rotaron. It looks like this. So there's a rod down the middle that's going to accelerate in terms of how fast it turns, and we see how long the mice can hold on for. If a mouse has a problem with motor function, with movement, then they're going to fall off quicker than a normal mouse. So this is the data, this is the open field. The white bars are the controls, and the black bars are those that lack microglia. I can't actually read that far, but it says this is the total distance moved, and you can see there's absolutely no difference between the two groups of animals. Over here we have the speed that the mice move on average, and you can see, again, no differences whatsoever. This is the rotor rod. The specifics aren't important, but you can see that both lines, the black and the grey, are pretty much the same as each other. So this again was very remarkable for us. Despite the fact these mice were lacking an entire tissue, for 2 months, they have no problems that we can detect in anxiety or in motor function. Of course, the important one is cognition, learning and memory. This is a very complex process that goes on inside our brains, and really we have to make sure that what we're doing is not disturbing this in these animals. So we have two measures again. This one here is known as Barnes maze, and it looks like this. It's a large circular platform with a series of holes all the way around the perimeter. One of these holes has an escape box underneath it. This is somewhere that the mouse wants to get to, because once he finds it, we'll take him back to his home cage. And so the idea is that every day we put the mouse on this platform, and then he's going to learn which one of these holes is the correct one, and each day he's going to get there a little bit faster if his memory works well. This one here is known as fear conditioning. It's a little bit different. What we do is we take our mouse and we put him in this box. This box has these wires going along the floor, and once we put them in there, we play a tone, a beep. After that, we give the mouse a very slight electric shock through the floor. And of course, the mouse doesn't like this. He jumps a little bit, and he finds it a little bit unpleasant. But then what we do is we take him a day later and put him back inside the same box. We then play the same tone, the same beep, and if the mouse remembers what happened to him the day before, he's going to freeze, he's going to panic. And we can measure how much time he freezes as a measure of how good his memory is. If the mouse doesn't have very good memory, he doesn't remember what happened after the beep yesterday, and he's got no problems with it. Okay, so I'm going to show you the data. Let's go through the Barnes maze first. Let's ignore this at the top. Okay, so the control mice are in grey at the top here. So on the first day, this is the Barnes maze, it took them over 100 seconds to find the right hole. The next day, they're a little bit faster. The third day, they were faster still. And by the fourth day, it was taking them about 70 seconds to be able to escape from that maze. But here's the incredible thing. These are the mice that have lacked microglia for 2 months. They actually find their way out of this maze faster. It doesn't take them the 100 seconds on day 2 that the normal mouse took. They can do it in 80, in 70. And so this was quite astounding to us. The mice that have no microglia, they don't have any deficits, and sometimes they even perform a little bit better. This was the fear conditioning test, and essentially we didn't see any improvements here, but once again we saw no differences. So this just reinforces the fact that despite the fact we are eliminating an entire tissue from the brain, this is not having any negative effects on these mice that we can measure in any way, shape, or form, and in some cases is even showing us beneficial effects. So from this, what we've shown is that we can give a drug, we can clear out all of these microglia from the brain. It takes between 7 and 21 days to actually get rid of them all, and actually nowadays we have new drugs that can do this in as little as 7 days. And from this, we've established that microglia are not necessary for any of the behavior or cognitive functions that we have looked at. And this means that we can now begin to directly study the role of these microglia in both the healthy, but also the diseased and injured brain. We know it's not going to screw things up on its own, so now we can start thinking about this as a therapeutic for these various different brain disorders. And so with that in mind, I'm now going to show you what happens if we apply this to models of Alzheimer's disease. Okay, so this is the research track. We don't do so much work on humans. Here we use mice and cells and test tubes and stuff like that. One of the tools that we have available to us are transgenic mice. These are mice that are genetically modified to develop Alzheimer's disease as they age. And so I showed you this picture earlier. These particular animals produce plaques, shown in the green, much like you find in the human brain of someone with Alzheimer's disease. And here, once again, is this really, really robust microglial response to these plaques. And of course, as these mice develop these pathologies, they also become cognitively impaired. They lose their memories in the same fashion that people with Alzheimer's disease do. And these are a very useful model to be able to look at potential therapeutics and determine what might be useful in Alzheimer's disease itself. Okay, so what we did is we took very old Alzheimer's disease mice. We actually took 23-month-old triple transgenic mice. These particular mice were developed here at UCI by Frank Lafurla, about 15 years ago now. And they developed both the plaques and the tangles that we see in the human brain. And we actually treated them for 3 whole months. So these are incredibly old mice, and we treated them for a very long period of time, relatively speaking. Okay, so the first question we actually had was, well, can we still get rid of microglia in the aged, inflamed brain? We knew that it worked in the healthy young mouse, but we had no idea if this would work in a situation where the microglia were old and a little bit unhappy. So here is a normal AD mouse. Each one of these green dots represents a microglia, and over here was one of the mice that was treated for 3 months. And you can see there has been a very, very significant reduction in the number of microglia. And if you count all these spots up, it's actually a 95% reduction of microglia. So this is great. The approach that we've worked out in these normal mice works in these Alzheimer's mice. We know that we can still eliminate all these microglia. We know that these microglia have been eliminated for 3 months. But of course, the key question is, well, is this beneficial? Is this good for the mice to no longer have these cells in the brain? And the answer is yes. When we test these mice on learning tasks, this one's actually known as a Morris Water maze. It's a very similar principle to the Barnes maze that I showed you earlier. We find that the untreated mice, they really don't learn their way out of this maze particularly well. But when they have no microglia, they learn it much, much better. And this is remarkable because these mice are so old and have so much pathology. Because these mice have not had microglia for 3 whole months, it also allows us to investigate questions about the role of these cells in the disease process. And one of the key questions that the field has is what the role of microglia are in the maintenance and formation of amyloid beta plaques. It's thought that perhaps they play a protective role and they stop them growing bigger or producing more of them. But here we have mice that have not had any microglia for 3 whole months, and we can count all of these plaques and we can measure the area of them, the size. And when we do this, we find absolutely no difference between the mice with microglia and those that do not have them. So this is the average number of plaques per field, and you can see that it's the same. Likewise, this is the average area of those plaques, and there's no difference. So this is really informative. It tells us that the microglia in the aged brain, despite the fact that they're surrounding the plaques and mounting this immune response, this inflammatory response, they're not helping us get rid of the plaques in any way, shape, or form. They're not affecting the plaque growth, they're not stopping new ones growing, they're basically doing nothing other than impairing cognition. And we can see same things for the tau pathology as well. We can look at what happens to the tangles, to the various forms of hyperphosphorylated tau that we see in these animals, and those without microglia have no changes. So again, it seems like the microglia in the old brain, they're not protecting us against pathology, they're not making us worse, but what they are doing is impairing cognition. Getting rid of these cells improves the phenotype of these animals. But of course, when you get rid of the microglia, you're doing beneficial things as well. You're eliminating from the brain all the things that these cells produce. And these include some of these cytokines that are implicated in, well, I see that. Okay, so for example, there are 2 cytokines, R1 beta and TNF alpha, which are heavily implicated in memory impairments, aging, and all these various disease processes. And when we eliminate microglia, we do see highly significant reductions in these cytotoxic cytokines, which is nice. So from this, we can tell that even microglia in the aged and diseased brains are fully dependent upon CSF1R signaling for their survival. We can achieve chronic microglial elimination in advanced Alzheimer's disease mice, and this elimination improves cognition but has no impact on pathology. So from this, perhaps treatment with these CSF1R inhibitors could represent a really useful therapy for Alzheimer's disease and perhaps other neuroinflammatory disorders as well. But I'm not finished there. One of the problems, and we've heard this earlier, is that there have been many, many different drugs tried in these same transgenic mouse models of Alzheimer's disease that have shown positive effects. They have then gone into the clinic, okay, into people with Alzheimer's disease, and they have failed, all of them, okay? And there have been tens of these drugs that people like myself and the other scientists in your audience have been convinced would work, you know, because they worked in these mice that we think modeled the disease so well, but as soon as we put them into humans, there's no effect. And so how can we be sure that when we're talking about drugs like this, we're not just going to find the same thing again? Well, when we look at mouse models of Alzheimer's disease, we have to take note that the fact that there are some fundamental differences between a mouse model of Alzheimer's disease and a human with Alzheimer's disease. And one of the key differences is the presence of neuronal loss. Humans have neuronal loss in abundance. The mouse models of Alzheimer's disease that we use do not. And the reason they don't is a question of time. The human brain, it has Alzheimer's for 10, 20 years. Those neurons are slowly dying off through the course of those years. The mouse only lives for 2 years. So it's not enough time for those neurons to die off in the same manner that they do in the human brain. So what we have done is we've taken the approach of using a second model of a mouse model, which recapitulates this extensive neuronal loss that you find in the Alzheimer's brain. So this is what it looks like. It's another transgenic. The details are not important. But what you need to know is that we can change the diet of these animals, and we can induce incredibly specific hippocampal neuronal loss. So these animals, this is what it looks like. These are the neurons in your hippocampus that form your memories. This is one of our mice that we have lesioned, and you can see that all of these neurons have gone away. These mice will lose about 80–90% of their hippocampal neurons, which is exactly the same as a person with advanced Alzheimer's disease. And so the thought is, well, if we can make mice with Alzheimer's disease pathology better, and we can make mice that have lost all of their hippocampal neurons better, then perhaps we have something that would be more promising for humans. Okay, so this is the makeup of the mouse. Don't worry about this unless you understand what this means, but it's a doc-soft system. So what we did is we took fairly oldish mice, adult mice, 5 to 8 months, and we induced this lesion that kills off about 80% of the neurons in the hippocampus. At the end of this, we put something magic back in the diet that stopped any more neurons from dying off. And then at this point, we treated them with this CSF1R inhibitor to see what effects it would have. So we have 4 groups here. We have a control group. We have one that is normal mice treated with the CSF1R inhibitor for a month. Then we have the lesion guys, and then the lesion guys who are then subsequently treated with the same CSF1R inhibitor. So this is what it looks like. This is the control group. In blue, we have all our neurons. So this is the neuron layer at the top. These are the neurons that form your memories. In green, we have our microglia. So you can see the cell bodies. They're nicely spread out throughout the brain. They look pretty normal. These are our mice that we treat with our CSF1R inhibitor for a month, and as I said earlier, it gets rid of all the microglia. They're pretty much all gone. And you can see that the neurons are still there. They're nice and happy. This has no negative effects on any of the neuronal populations. These guys have had the lesion. Pretty big difference. The first thing to note is actually the hippocampus is now smaller. It's shrunk because there's been such enormous neuronal loss, the same that you find in Alzheimer's disease. This is the cell layer here. Look how much thinner this is than the previous slide. This is what's happened. These mice have lost 80% of their hippocampal neurons. And you can see that this neuronal loss has elicited a massive inflammatory response, which is mediated by these microglia. You can see they're activated. You can see that they're more numerous. And so the question is, okay, well, can we get rid of these cells, and can that facilitate functional recovery? So this is what it looks like when you have a lesion and then you treat with these CSF1R inhibitors. So again, the neuron layer is still thin. We killed off the neurons before we administered this drug. You can't bring back neurons that are dead. But you can see we've had a very dramatic effect on the number of microglia that are present. We've gone from this to this. So once again, these microglia, despite being highly activated, are still fully dependent upon this CSF1R signaling pathway. This is just a quantification. We find with the lesion, you actually double the amount of microglia, and that with our drugs, we can basically get rid of pretty much all of them. So the key question is, okay, well, is this beneficial? Does getting rid of these microglia facilitate functional recovery, which is basically what we're interested in at the end of the day? And I'll tell you that it does. This is the elevated plasmase, and you'll remember we said that when a mouse is not performing properly, it spends more time out in the open and less time in the closed arms. Well, in red we have our lesioned animals, and you can see that they're spending less time in the, which one was that? In these arms, in the closed arms, and more time out in the exposed arms than the controls. But when you have eliminated microglia afterwards, you can see that we get a recovery. So it's not 100%. I mean, there's no way that we can make up for the fact that we've killed off 90% of the neurons, but by eliminating the microglia, we have facilitated some recovery. This is the Morris water maze, same thing that I showed you before. This group in red at the end, these are the lesion mice, and you can see that all the other three groups are performing a little bit better. This is a probe trial. This is a memory test that is done at the end of the experiment. It just tests how well the memory of these animals is. The lower the number the lower the number, the better. And what we find is both the mice treated with the drug that lack microglia or those lesioned and then treated perform incredibly well. They perform better than the controls and the lesion guys. So elimination of microglia not only improves memory just in healthy mice, but is able to functionally improve mice with a pretty severe lesion as well. So just because time's running out, this is the last slide. When we look at what microglia might be doing in these brains to impair functional recovery, we can see that they're doing things to synaptic proteins. When we eliminate microglia, we end up with increases in synaptic puncta. These are the connections that neurons form between each other that allows them to communicate. Likewise, when we lesion, we see that these puncta become enlarged and dysfunctional, and that when we remove the microglia, this restores things back to normal. Okay, so to conclude, what we've shown is that microglia in any activation state are still dependent upon signaling through the CSF1R signaling pathway for their survival, and that elimination of microglia following this neuronal lesion facilitates both functional recovery but also synaptic alterations. So what I've shown is that we get good functional recovery in both models of Alzheimer's disease and extensive neuronal loss, and that therefore together this provides a compelling argument that these should be developed further, not only for Alzheimer's disease but perhaps for other neuroinflammatory diseases as well. These are the people who actually did the work. There's a few of them here in the audience today. Frank Lafurla made the mouse models that I've discussed today, both the triple transgenic and that neat neuronal loss model. Brian West at Plexicom's my drug dealer, who gives me these wonderful drugs to do things with, and funding sources. And with that, I would be happy to take any questions. Thank you.